Okay, I think all clocks are a little different, but mine says it's on the hour. And I am going to get started. I know that there will be others joining. I do want to welcome you all on the phone to our research update series, which we hold monthly. And the goal is really to bring to you, as patients, families, chapters, ALS community, the latest on research. Many of the presentations are work that the association funds. Sometimes it's more clinically focused. Sometimes it's more basic science. We always record the sessions and we post lay summaries, so I understand you can't perhaps make it every month, but be assured that we have materials for you to review. The presentation will be around 30 minutes, and then you'll have the opportunity to ask questions, and I encourage you to please do that through the chat to research the research icon. It is my distinct pleasure to welcome today our speaker, Dr. Steve Burden. He is the professor at the Department of Cell Biology and Biochemistry at New York University Langone Medical Center. He really is a tremendous asset to the ALS field. He comes in with developmental neurobiologist background, has focused his research in the lab on the neuromuscular junction, and I thought it was so exciting to have him present because he represents a component of our research programs that we haven't featured often. As you all know, the association funds a variety of programs, from postdoctoral fellowships to small basic research studies, and over the years we have encouraged studies that really move things from the laboratory into the treatment phase. So these are our translational research programs. And in this case, Dr. Burden is being funded through a contract program where we encourage partnership with academia and industry to take a very novel approach in the laboratory and to help bring it into a treatment mode. So we're very excited to have Dr. Burden, who has such a specialist and expertise in an area that's so important, the junction between the motor neuron and the muscle, and he will illustrate today how this might be a potential area of treatment. So over to you, Dr. Burden. Thank you very much for taking the time today.
Very good. Thank you very much, Lucy. So it's a pleasure for me to be here today and to talk to you. I understand you've had a busy day, that you've had a talk earlier this morning from Bob Brown. I am, as Lucy said, a basic scientist who has studied for a long time how neuromuscular synapses formed and how once formed these synapses are maintained and stabilized. And as you well know, in ALS, the stability of this synapse becomes compromised, and I'll tell you our perspective on the ways in which it first becomes compromised, and we believe that our fundamental studies in trying to understand how synapses form and are stabilized have led us to an unusual insight into the causes of ALS and ways in which ALS might be therapeutically treated. So trying in there with me, I think I'm going to go through a lot of basic science. I'm going to try to explain it in the clearest possible fashion. There will be times when the terms I use are new and novel, and you might not grasp them when I first mention them, that I'm going to repeat the names of certain molecules or structures again and again. So I think by the end of the 30 minutes, you'll be somewhat familiar with the terms and the concepts and the ways in which we're trying to develop a therapy to treat ALS and our particular perspective on this disease. So what you're seeing in the first image is a muscle that's innervated by nerves that are forming synapses, connections with the muscle. And I'll go through some of the structures that are illustrated in this colored slide, and that'll be an important part of the theme that I'm going to develop over the next 30 minutes. So all the pictures you see are from a mouse neuromuscular synapse. And you'll have to believe me that most of what we know about synapses that form and work in humans come from studies in model organisms, many of which are studies that come from the mouse. And the mouse nerve muscle synapse resembles in many ways both how it appears and how it functions human neuromuscular synapses. And as I suspect you've heard in the past, and I'll emphasize again today, that there's a mouse model for human ALS, and it's generated by genetically altering the mouse so it carries a mutation in one of the genes that's known to cause ALS in humans. And that's the superoxide dismutation, or abbreviated as SOD1. And this carries a particular mutation in one amino acid, a glycine that's at position 93, that in humans and in the mouse model has been converted to a different amino acid, alanine. And it renders the protein different, and different in a way that results in ALS. So this is a view of the mouse neuromuscular junction, and in blue, you can see these long, thin stripes of cells that are sitting one by one another, side by side, and those are muscle cells, very, very long cells that you're seeing just a portion of the muscles here. The muscle cells are a very long cell. In the mouse, this particular muscle is about 1 centimeter long. In humans, of course, the muscle cells from knee to hip could be nearly 1 meter long, or from ankle to knee could be over 1 meter long. So these are very, very long cells, and these long cells normally are quiescent. They don't contract, they don't move, unless the nerve provides a signal to the muscle cell that triggers it to contract. So the nerve controls the contraction of the muscle. And the nerves we can see here, because they're stained and appear green in the image here. So let me see, can you see the arrow here that I'm drawing over the nerve? So this is,
Steve, I don't see it.
You don't see that. Okay, so I'll just continue to describe. So the green structures you see in the middle, they're bundles of individual axons, and those nerves arise from the spinal cord. That's where your motor neurons are located. The axons leave the spinal cord. They go out to the periphery to all the muscles, whether they're the muscles in your fingertips, the muscles in your arms, the muscles in your hips, the muscles in your toes, the muscles in your heads. So the axons have to travel very long ways to reach the muscle. So these axons are bundled together, and running from the lower left to the upper right. And then they branch. You can see the small little green branches emanating from that bundle of nerves. And then they stop adjacent to that main nerve, somewhere in the middle of the muscle. And what's highlighted in the yellow box, and then to the right, in a higher magnification view, you can see 3 axons, 2 that are continuing to grow from right to left, and 1 that makes a small bend up toward the top in the inset, and then branches in this beautiful flower-like structure, and that is the synapse. That's the place where the motor axon stops, makes a connection with the muscle, and communicates with the muscle. And I'll show you in other slides the further detail of that connection. So that's the place where your nerve communicates with the muscle. The way it does so is it releases a small molecule, acetylcholine, referred to as the neurotransmitter, and it's going to leave the nerve terminal and then diffuse across the space between the nerve and the muscle, and then bind to neurotransmitter receptors, those that bind acetylcholine, so they're called acetylcholine receptors. And when the nerve acetylcholine binds to the muscle acetylcholine receptors, that initiates a process that ultimately leads in contraction of the muscle. So that's the way the nerve controls movement of the muscle, by controlling contraction by acetylcholine. So what you're seeing in that image, that inset, is the green axons terminating on the muscle, and the acetylcholine receptors we can visualize in the microscope as well, and those appear as the red stain in that inset, and also in the lower power image that's the basis of the slide to the left. So what you can see is the nerve is terminating in this beautiful flower-like structure, and right underneath where the nerve is located are a very high concentration of the neurotransmitter receptor, acetylcholine receptors in the muscle. And it's essential that they be found at high concentration there in order for the nerve to stimulate a large enough response in the muscle to generate contraction. Let me see if we can move on to the next slide here. There we go. Okay, so now let's talk about, I'm going to describe to you our perspective of ALS in 2 slides, and then I'm going to come back to that neuromuscular synapse that I just introduced to you in the previous slide. So you're all devastating a disease, ALS is, and you mostly probably hear it described as a neurodegenerative disorder. And indeed it is, and it's a neurodegenerative disorder because the motor neurons that are sending these axons out to the muscle to form synapses die. And when these neurons die, the neuromuscular synapse can no longer function because there's no longer a connection between the nerve and the muscle. But well before the motor neurons themselves die, there's a loss of a connection between the nerve and the muscle, the very connection which I just illustrated for you in the previous slide. And we'll see more detail of this in the next few slides. But I'm just going to describe here that it's a progressive disease which begins by a disassembly of the neuromuscular synapse, the structure you just saw, and ultimately leads to motor neuron cell death, which leads to lethal respiratory paralysis, largely, although not always, without cognitive impairment, and as you well know, within just a few years of diagnosis. The incidence of the disease, as you probably heard from Bob Brown earlier today, is around 1 to 5 and 1 to 10,000, and that's not so terribly different from other well-known neuromuscular diseases, muscular dystrophy, and autoimmune myasthenia gravis. You probably also heard from Bob Brown earlier today some of the causes of ALS. And there are genes, I earlier mentioned superoxide dismutase, or SOD1, where mutations of that gene can result in ALS. And there are other genes, TDP43, FUS, and more recently discovered this chromosome 9 gene called C9 or 72. Mutations in that are also responsible for familial forms of ALS. Bob, I'm sure, mentioned to you, and as many of you know, not all forms of ALS are known to be caused by mutations in single genes. In fact, probably less than 20% of all cases of ALS can be explained by mutations such as those in SOD1, which are single gene mutations. More of them appear as sporadic mutations in which the cause is not entirely clear at the moment. But it's nuts. In all of these, as illustrated, as I mentioned here, 4 genetic causes of ALS, as well as all of the sporadic forms of ALS, the progression of the disease and the course of the disease, although variable from patient to patient, is not dependent upon whether it's a mutation in SOD1 or TDP43 or sporadic. They all appear to progress in a very similar manner, which I already described, and that is the withdrawal of the motor axons from the muscle is the first sign of disease, and that ultimately leads to this progressive loss of motor function that results in lethal paralysis. And also well know there are no real therapies to cure, let alone effectively slow disease progression. Our perspective on this disease is that although most people study, as I mentioned, the loss of the motor neuron, the cell death that's associated with ALS, our interest for a long time has been in what are the nature of the molecules and signals that keep the nerve attached to the muscle and allow the nerve to signal to the muscle and the muscle to signal back to the nerve to keep that synapse attached. And so we've been very interested in the fact that the withdrawal of motor axons from the muscle is the first sign of disease in ALS. And to us, this seems very, very important, because if that occurs, yet you can find a therapy to keep the motor neurons alive, but you can't keep the synapse attached, you haven't accomplished very much, because you need to have that nerve attached to the muscle in order for the synapse to function. That has motor neurons intact but without connections to the muscle can't function. So we think this is really very, very central to the disease. And then in the next slide, in the mouse, but this was the same kind of phenomenon you see in humans. This is an example of a mouse that has a mutation in SOD1, so we'll call it our mouse model or our ALS mouse. You can see there are 3 synapses in this image, and the lower one, labeled A3, looks very much like a normal synapse, which you can see the axon in green terminating on the muscle in a very complex branching pattern, and underneath those nerve terminals are the acetylcholine receptors labeled in red, so you get a yellow mixed merged image of the axon on top of the acetylcholine receptors. In the A1, you can see, as it was marked by the star, the motor axon is pulled off or withdrawn from the synapse, still marked by A1, where the acetylcholine receptors are still found in the muscle. So that muscle fiber is what's referred to as denervated. The nerve was attached, overlaid like in A3, but the nerve has now retracted from the synapse, leaving the muscle denervated and no longer functional. It's kind of the fact that this is a complex disease, because in this particular synapse, which the authors did, is they imaged it as with time over the period of several days to weeks. And what they found is it progressed initially from A2, started like A3, normally innervated, then the synapse pulled off, it looked like A1, and then axon flew out and extended and is beginning to make contact with A2, a process called reinnervation. So in A3, it illustrates normal innervation, in A1, denervation, and A2, reinnervation. Now, if the reinnervation was really effective and permanent, you would recover or maintain function. The problem is the reinnervation is not permanent. Eventually, all of the synapses will look like A1, and at that point, there'll be complete paralysis. Okay, so let me move on. So this is the phenomenon we're interested in. Why do the nerves pull off the muscle, and can we find a way to either keep them attached or keep them attached for a longer period of time than they would ordinarily be in this disease setting? Now let me move on to a summary of the types of studies that my lab and other labs have carried out over the past dozen or more years to try to understand what is the nature of the signaling between the nerve and the muscle that normally keeps the muscle cell differentiated to make sure it has all the right molecules it needs in order for the nerve to trigger contraction of the muscle, and for the muscle to signal back to the nerve to make sure it's releasing the neurotransmitter and keeping that synapse attached. And we believe that now, having a better understanding of what are the normal signals to keep the synapse attached gives us insight and potential therapies to try to keep synapses attached more strongly when they're compromised, as they are in ALS. So this illustrates some of the nomenclature and some of the names of molecules, which I'll mention again and again. They'll become more and more familiar to you over the next 20 minutes or so. So the nerve terminal is shown up above. So it has an axon and a bulbous-like nerve ending. And it shows little round structures, shown 4 of them in white. And these are structures that contain the neurotransmitter labeled to the right, ACH or acetylcholine. So that's the molecule that's going to leave the nerve terminal, and then it's going to bind, shown on the far right in red, labeled ACHR. So that's the neurotransmitter acetylcholine receptor. So once acetylcholine leaves the nerve terminal and diffuses over to those acetylcholine receptors, that's the mechanism by which the nerve is going to elicit contraction of the muscle. Now, in order to get those acetylcholine receptors at high concentration in the muscle membrane at the synapse so it can respond in a robust manner to the neurotransmitter, there are a lot of other molecules that need to work in the muscle. So this molecule that's labeled agrin and is illustrated in blue, that's a molecule that, like acetylcholine, is made by the nerves. So it's made by nerves, it's released by the nerves, and then it binds to a molecule that's found in the muscle membrane. And that molecule is called LRP4, and it's multicolored. It's yellow, green, and blue. And what agrin does when it leaves the nerve terminal, it binds to LRP4 in the muscle, kind of like sticky paper. So it's like a fly binding sticky paper. But when it does so, it doesn't just attach. By ways that we still don't understand. We don't understand this machinery in all its detail, but we know once agrin binds LRP4, then it stimulates association between the molecule LRP4 itself and the purple molecule that's sitting next to it, a molecule called MuSK. Now, MuSK is a very important molecule because when LRP4 binds MuSK, this is an enzyme, and this enzyme is inactive before LRP4 binds to it, but once LRP4 binds to it, it stimulates this enzyme, and this enzyme transfers phosphate molecules onto other molecules. So it serves as a switch So without phosphate, molecules are inactive or silent, and when they have phosphate attached to them, they become active. So agarine binds LRP4, which binds to and stimulates MuSK, which stimulates the transfer of phosphate onto a series of molecules, and through a series of reactions by transferring those phosphate molecules, that ultimately leads to this clustering of the red acetylcholine receptors at the site where MuSK is activated, which is that.
So just to summarize again, that agarine is released from the nerve terminal. It binds to LRP4 in the muscle. That stimulates association with MuSK, this kinase, this enzyme that transfers phosphate onto other molecules and is essential for aggregating acetylcholine receptors at the synapse to make sure that the nerve can control the contraction of the muscle. So that's how the nerve signals to the muscle. But as it turns out, the muscle signals back to the nerve to keep it attached. That's an important step, and as you've seen, that goes awry in ALS. So if we move to the next slide, so it turns out that two of the molecules that you've just heard about and learned about, LRP4 and MuSK, remember they're receiving a signal from the nerve and stimulating specialization in the muscle by accumulating acetylcholine receptors. But it turns out that both of these molecules have a dual function. They function not only in signaling from nerve to muscle, but they're also required for signaling back from muscle back to nerve. And this was an important discovery that's made in the past half dozen or so years. And an illustration of their role in the signaling from muscle back to nerve is shown in the next slide. On the left-hand side is this image very similar to what we saw at the very beginning. And wild type refers to normal. So this is a normal mouse muscle. And in the green image in the upper left, again, you can see the axons from the motor neurons in the muscle bundled together and then branching in this tree-like structure right adjacent to, just above, where the main nerves are bundled. And then they stop there, and they form synapses. So in the lower image, we're looking at the same muscle, but now instead of looking at where the nerves are, we're looking where these acetylcholine receptors are that are going to respond to acetylcholine released by the nerves. And as you can see, they're concentrated in a band in the middle of the muscle, right underneath where those nerve endings are. That we already saw before. Now on the right-hand side is a muscle from a mouse that carries a mutant form of MuSK. So MuSK is unable to work well in the mouse to the right. And as you can see, there are two very striking differences, both in the red and the green images that you see when you lack MuSK. So in the lower right, in red, you can see that there are no clusters or aggregates of acetylcholine receptors in the muscle. And that's because, as I told you, when MuSK gets activated in the muscle, it stimulates the clustering of acetylcholine receptors in the muscle. So if MuSK can't function, acetylcholine receptors can't cluster and aggregate, and there's a uniform expression of these receptors in the muscle. So that means the synapse isn't going to work very well. And now you already saw in the cartoon that MuSK is in the muscle cell, and the way the nerve communicates with MuSK or regulates it is it binds to LRP4, which then binds to MuSK and stimulates it. But you can see that the nerves in the upper right, they're not in the right position in a MuSK mutant as well. So the defects are not just in the muscle, the lower right, they're in the nerves as well, the upper right. So the nerves are in the muscle, they're in the right position, but instead of stopping and differentiating, they just grow all over the muscle, up and down the muscle, without forming a synapse. So this tells us that MuSK normally functions in the muscle to stimulate the muscle specialization, the anchoring and clustering of acetylcholine receptors, but it's required for the signals to go from muscle back to nerve to tell the nerves to stop and to become specialized. And in the next slide, you can see that the same kind of defects are found in a muscle from a mouse that lacks LRP4, the molecule that binds agarine and stimulates MuSK. So on the right, again, you can see the acetylcholine receptors are uniform in the muscle. They're not aggregated. And on the left, you can see the nerves, instead of stopping right adjacent to the bundle of axons in the middle of the muscle, like in the MuSK mutants, they grow all over the muscle. So these data and a lot of other experiments have led to a model shown in the next slide about how these molecules work. So let's focus first on the far right, which is what we've been talking about. So NT, the nerve terminal, releases agrin, shown in as a blue-colored molecule, which, as we mentioned earlier, binds to LRP4, and then, as you can see from the arrow, from LRP4 to MuSK stimulates MuSK, and that stimulates all the specializations on the muscle side, shown by the curved arrow below MuSK. But in the middle panel, as well as on the panel to the right, we're illustrating this additional function of LRP4 MuSK. So you can see arrows going from LRP4 back to the nerve terminal. And that's meant to illustrate that the signaling between LRP4 and MuSK on the muscle side signals not just within the muscle, but also back to the nerve, and is responsible for the nerves to stop there and differentiate. And if LRP4 and MuSK don't signal well, then both of those, all the curved arrows are missing. The curved arrow in the muscle, no muscle differentiation, as well as no differentiation of NT, the nerve terminal. They won't stop there, they grow all over. So if we put the next slide up, so this idea, this knowledge of knowing that LRP4 and MuSK signal, as shown here in this slide in the red arrow, back from the muscle, back to the nerve, led us to consider the idea that if LRP4 and MuSK are normally signaling to keep the nerve attached to the muscle, we wondered whether if we could find a trick, a way to make this what we call as retrograde, or signaling from muscle back to nerve, stronger to increase the signaling, whether we might be able to keep synapses attached that were otherwise compromised for any of a variety of reasons. If the nerves were weakened in their attachment to muscle, if we could trick the muscle to increase the amount of signaling coming from muscle back to nerve, could we keep the synapses attached? So in the next slide, let's see, this is going to work. We tried to do exactly that. So indeed, we had a trick, and the trick is illustrated, the way we describe it here is it's called HSA-MuSK-L. And it's a way in which we can genetically alter MuSK to make it a little more active. And this involves genetic manipulation. So we have to introduce a gene by genetic trickery into the mouse to make MuSK a little bit better as an enzyme, a little stronger than it would be otherwise. And so on the left image, WT refers to normal, and this is a normal mouse muscle in which we're simultaneously staining for nerve endings and acetylcholine receptors. So the overlap of yellow and red produces a green-like image, and you can see these beautiful 1, 2, 3, 4, 5, 6 or so flower-like structures in the middle of the muscle that are green. So there's perfect overlap of where the nerve terminals overlie where the acetylcholine receptors are in the muscle. And this is a mouse that's about 140 days old, so it's almost 5 months old. On the middle panel, the one to the right here, it's labeled HSA-MuSK-L. That's where we've a muscle from a mouse in which we've applied some genetic trickery, and so there's a little more MuSK. It's a little bit more active, a little bit more functional than it would be normally, and the synapses, they look perfectly normal. There's no harm done here, no negative effect on synapses by making MuSK a little bit more active. In the next image on the same slide, you'll see an example of the mouse that you saw earlier. So this is from the SOD mutation, which resembles that found in humans, that causes this mouse to undergo a pathological phenotype similar to what's seen in ALS in humans. And now at 140 days, these synapses, more than half of them are all red. And they're all red because the acetylcholine receptors are still there, but the nerves have pulled off. There's no yellow over there, so there's no green. There's no overlap of the nerve terminals in the muscle. So these are naked or denervated synapses. The nerves have been pulled off. So now what we did is we said, okay, let's take this SOD mouse in which the nerves have pulled off, and let's apply our genetic trickery here. Let's make MuSK a little bit more active in this mouse and see if we can keep the synapses attached better if we've just increased the retrograde signaling from muscle back to nerve. And the next image shows you that mouse. So indeed, that is the case. If we increase MuSK activity in this mouse model of ALS, this SOD mutation, you can just see in these images that most of these synapses have yellow on top of red, and so they appear red. Appear green. So in the lower left, there's a synapse that's nearly all red, so indeed that's denervated. We haven't been able to help that synapse. But the other three synapses in the muscle are innervated now, whereas otherwise, if we hadn't increased MuSK, they would look like those illustrated just in the SOD1. They would be all red. So if we go to the next slide, you can see we've been able to quantitate this effect. So the images that you saw in the previous slide are illustrated once again in the panels at the bottom part of the slide. And now we show in a graph what percentage of the synapses become denervated or devoid of nerve endings, of nerve terminals over them with time in the mouse. And in red, let's start with red, is we're looking at the ALS mouse or the SOD mutant mouse. And as you can see, at about 110 days after birth, it's pretty normal. Nearly all of the synapses are innervated. None of them are denervated. Whereas at 120 days, almost 20% are denervated, and by 140 days, over 50% are denervated. And that stays relatively constant over the 20 days. In the normal mouse, that's shown in green. As you can see, the synapses remain normally innervated as far as we've looked, so up to 180 days, and that'll stay that way for up to 2 years. But importantly, if we increase MuSK activity by this genetic trickery in the SOD mouse, you see the data shown in the purple or blue line there. So first of all, you see that at 120 days in the SOD mutant mouse, although 20% of the synapses are denervated, virtually none of the synapses are denervated if we increase MuSK in this SOD mutant background. And over the next 40 days, up through 150 days, there's a dramatic reduction in the amount of denervation simply by increasing MuSK activity by this genetic trickery. And for example, on the panel to the right, we were just showing you the data at 140 days. And again, about 53% of the synapses are denervated in the SOD mutant at that time, and only 8% if we increase MuSK activity. So by increasing MuSK, increasing the power of this enzyme or the activity of this enzyme, we can preserve the synapses, keep them intact for a much longer time than they would be otherwise in the SOD mutant background. So in the next slide, I'm going to give you a little bit more details about the science. So I've described synapses as, if you look in the three images at the lower part of the panel, I described two as innervated, and I've subdivided those as normally innervated. These are perfectly normal synapses. And then to the right, or the middle panel, is a synapse that's innervated, but it's only partially innervated. And then on the right are synapses that are totally denervated. So that's the way things progress, is they progress from perfectly normally innervated, and then the nerves gradually pull off the muscle. So initially they're partially innervated, partially denervated, and then fully denervated. And in the previous slide, the way we quantitated everything is we just measured the far right. How many were fully denervated? Now if we move to, again, forward here, one slide, now we're going to measure how many, in the next slide, how many of the slides are really perfectly innervated, like the ones on the far left, normally innervated. Next slide. Okay, so this is normal innervation. So again, the normal mouse, shown in green, they stay normally innervated for a very, very long time, up to 160, by 160 days. Actually, some of them are starting to become already fragmented. That's actually an old mouse, and so that's at 160 days. But importantly, for the ALS mouse, in red, you can see that by 110 days, already only 80% of the synapses are perfectly normally innervated. Even then, 20% of the synapses are already partially denervated. And normal innervation falls off dramatically, so that by 140, 150 days, only 20% of the synapses are normally innervated. And those, so most of the synapses are either partially innervated or fully denervated. And by increasing MuSK activity, like you saw earlier, If we look only at normal innervation, by increasing MuSK activity, the purple line shows you that we can keep these synapses looking very, very normal for about 40 days, where they would otherwise start to fragment and become denervated. So the question now is, if we can keep these synapses morphologically, the nerves attached to the muscle, it was possible that although we kept them attached, maybe the nerves were so compromised because of this altered form of SOD1 that although they're physically there, they just wouldn't work. And so the muscle wouldn't contract and the mouse wouldn't improve in its motor dysfunction. Alternatively, it's possible that keeping the nerves attached to the muscle would allow the synapse to actually work and allow the mice to improve their motor behavior. So we tested that, and that's shown in the next slide. So this is actually a pretty crude assay. What we're measuring is how well, when we take a mouse and ask it to hang on to a bar, how long can it hold on to the bar? So this is one measure of grip strength. And as you can see in green, again, the wild type, it can stay attached to the bar for a very, very long time, and it's continually strong up to 170 days after birth. In red, you see the ALS mouse. So again, at about 120 days, when the synapses are starting to pull off from the muscle, well before the motor neurons have died, they're not going to die until about 180 days. Well before they've died, they're starting to lose motor strength. So losing that attachment of the nerve to the muscle is really critical in losing motor function. And by 140 days, it only has 10% of its normal strength. By increasing MuSK the enzyme activity, again, in this SOD mutant background, you see the purple line. So at all times from 120 days on, there's much improved strength by keeping MuSK a little more active. So we've not only, by increasing MuSK activity, not only are the synapses morphologically connected to the muscle, but they actually work and improve motor behavior. So we think this is very, very important. Okay, next slide. So although we've improved motor function over this long period of time, the time between 110 days and 150 days, you can see the difference between the red and the purple. But by 160 days, even though we've increased MuSK activity, we can't permanently keep the synapses intact. Eventually, they do pull off, and eventually the mouse does lose its motor function. So by increasing MuSK activity, this is a way of delaying the onset of the disease symptoms, decreasing the severity of symptoms, but not preventing disease symptoms from ultimately arising. And if we look at the next slide, if we look at the longevity of these mice, because they lose their synapse of the muscle and they no longer can function, then these mice die of respiratory failure. And the time at which they die is shown in the red curve. The wild-type mouse is shown in the green. And as you can see in yellow, if we increase MuSK activity in this mouse model, we see what's shown in the yellow line. So there is a slight increase, but very, very modest, in their survival of the mice. And that's due to the fact that ultimately the synapses do pull off. So there's a window of opportunity when we can dramatically increase muscle function, but it's not permanent. In the next slide, we have a summary cartoon of what we believe we found, and I'll give you an explanation of both why we think the effect is temporary, not permanent, though potent, and then how we think we can use this knowledge to try to design therapies that can be used not just in a mouse, but in humans. So again, in this cartoon, the wild type or normal mouse is shown on the far left. Again, the nerve signals to the muscle to keep the muscle specialized, but there's a back signal from the muscle to the nerve, which we call an attachment signal. That requires MuSK, this kinase in the muscle. In the ALS mouse, we believe that there's defects in the motor neuron that lead to a decrease in this attachment signal from the nerve to the muscle, or the ability of the attachment signal from the muscle to be recognized by the nerve, and the nerves pull off. And then we believe, shown in the far right, by taking this this mouse carrying a mutant form of SOD1, by improving or increasing the activity of this enzyme in the muscle, we can keep the neurons attached to the muscle longer by increasing the amount of the signal that's coming normally back from the muscle to the nerve. So in the next slide, we provide some sort of rationale for what's going wrong in our view, the way we think about this attachment signal, and why we think the effect is temporary, not permanent. So the model is that in a young mouse in which the synapses are normally attached, which is a P100, the SOD mutation is already having an effect, but it's not visible. The nerves are still attached to the muscle. The mouse still has normal motor behavior. And again, there's this retrograde signal from the muscle going back to the nerve. And the nerves, in order to sense and respond to and listen to this signal from the muscle, they must have a receptor for it. They must have something that binds this signal from the muscle to respond to it. And that signal, shown in dark blue, we are currently trying to identify. And that will be a very important step in trying to understand the signaling mechanisms that keep nerves attached to the muscle. But the model is that in an ALS mouse, or in any of the sporadic forms of ALS where the nerve is compromised, many things change in the motor neuron. Among the things that change in the motor neuron are proteins normally that are made in the cell body, in the spinal cord, need to be transported or shipped down this very long axon. Remember, the neurons are in your spinal cord, and some synapses can be in your toes. And so proteins, the machinery for the way in which that nerve terminal works, need to be transported this long distance. And it's known that in mutations that cause ALS, that transport process doesn't work as well as it should. And so we hypothesize that among the proteins that need to be transported from the cell body down to the terminal is this dark blue receptor that the nerves use to hear the signals that are provided by the muscle. And so we believe that as they get less and less of this receptor, this blue signal in the nerve terminals, they can no longer hear and respond to the signals the muscle are making. And therefore, they pull off and withdraw. As you saw earlier, when a mouse lacks LRP4, lacks MuSK, and can't provide the signals, the nerves grow all over the muscle. They can't hear the muscle. So in the next slide, what we've done, it shows kind of the diagram of the experiment I just described. What we did is we increased retrograde signaling in the muscle. So there's more of this yellow-green molecule that's made by the muscle. On the left, you can see I've shown one such molecule. On the right, by increasing MuSK expression activity in the muscle, we now have, I'm showing here, 3 molecules. So we've increased the retrograde signal. Now what's going on in the nerve, we haven't changed. So it's compromised, it's becoming defective in transport, so it's providing fewer blue molecules shipped down to the nerve terminal. But because we've provided more signal from the muscle, even when there are few blue molecules, like in the middle, just one, whereas in the left-hand side, in a normal retrograde signaling situation, that's not enough to keep it attached. But if we give more and more of the retrograde signal from the muscle, as shown on the right, we think we can keep that receptor engaged and keep the synapse stabilized. And that's this intermediate stage around P140 when we think that's the mechanism by which the nerves remain attached to the muscle and we can improve motor function. But of course, at the same time, the nerve is deteriorating. And eventually, there's going to be less and less of blue, or the receptor, the molecules for hearing the signals from the muscle. And eventually, there's going to be so little, despite all this retrograde signal that we're providing for the muscle, the nerve's just not going to be able to hear, and eventually it will pull off. So that's the way in which we think about this. Now let's move on to how we can nonetheless use this information to try to understand how we can use this insight to develop therapies for ALS. So this summarizes the key results from the studies, is that an increase in the MuSK activity in the ALS mice maintains attachment of nerve terminals to the muscle for an extra 40 days, which in this mouse that only lives 160 days is about 25% of its lifetime. And that improved attachment of the nerve to the muscle improves motor function. So in the next slide, this leads to what you would like to do is find a way other than genetic trickery, which would be difficult in humans, have the same effect, to increase the activity of MuSK in muscle tissue of an ALS patient, and ask whether if I can increase MuSK activity by pharmaceutical reagents or other types of reagents, might that have the same effect that we've seen here in the mouse, keep the nerves attached to the muscle longer, improve motor function for a longer period of time, and improve the quality of life for the patient and family. And so I've listed here 4 ways in which I can, on paper, conceptualize ways in which we could stimulate MuSK activity, to increase the activity of this enzyme in normal tissue. And I'm going to focus in the last slide on the last mechanism here, which I refer to as activating antibodies to MuSK. So the way I've described the normal biology here is that the normal way in which MuSK is stimulated is this molecule from the nerve. Actually, maybe we can go back a slide. This molecule agrin that's made by the nerves binds to LRP4, which then binds MuSK and then stimulates MuSK. So in fact, you could come up with a strategy knowing that for stimulating MuSK. Since agrin normally binds LRP4, which stimulates MuSK, maybe you could just supply a little more agrin, and maybe that would stimulate MuSK. And that's a reasonable suggestion, and that's the number one idea listed here. The other idea is, again, remember, the way in which agrin stimulates MuSK is it binds LRP4, which then binds MuSK and stimulates it. So maybe you wouldn't need agrin. Maybe there's a way in which you could supply LRP4 that would now bind MuSK and stimulate MuSK. And so, of course, that's a reasonable strategy as well. A third way is referred to as small molecules that selectively activate MuSK. So you could screen for drugs. So pharmaceutical companies do this all the time. They have a large cache of drugs in their profile, and you could screen without knowing much about how they act, and just look for drugs that are able to stimulate the activity of this enzyme. And that's a reasonable way to try to find a way to stimulate MuSK in an animal in vivo. Now, the fourth is a way in which you could stimulate MuSK by a completely different route. So some of you may be familiar with antibodies that are used therapeutically to inhibit activities of molecules. So, for example, you might know in inflammatory disease and rheumatoid arthritis, that there are ways in which you can dampen or decrease the activity of the immune system by using antibodies that block the function of molecules that are inflammatory. You can also, there are cases in which there are kinases that resemble in some ways MuSK, that when they become too active, these other kinases, they can actually cause cancer. And there are antibodies that are used to inhibit the activity of those enzymes, so they're less active and therefore are therapeutic. But there are times when you want, instead of to dampen the activity of enzymes and molecules, you want to increase their activity. And that's what we want to do here in this case of MuSK. We want to increase its activity in muscle. And there are ways in which antibodies can have, instead of an inhibitory function, an activating function, and actually stimulate rather than antagonize the function of molecules. And so in the next slide is a study that was published a long time ago in 1997, and this was a study carried out by a very powerful and effective biotechnology company in South San Francisco called Genentech. And they were working on this kinase MuSK as well for a short period of time. And they had identified in their study a series of antibodies that bound to and recognize MuSK in a way that's, in a sense, similar to the way LRP4 recognizes MuSK. And most of those antibodies that recognize MuSK did nothing to MuSK. They bound it, but they didn't inhibit the enzyme, and they didn't stimulate the enzyme. But two of the antibodies that they generated had a very unusual activity, and they stimulated the activity of the enzyme. And so we've begun a collaboration with Genentech to try to study these molecules in more detail and find out whether they can have this activity and stimulate the kinase in vivo first in a mouse. And we've begun first by analyzing these antibodies in a more simple setting, which is shown in the next slide, in which we take muscle cells from a mouse and we grow them in a tissue culture dish out of the animal in plastic in the laboratory setting. And we're able to measure how active MuSK is as an enzyme. And in the upper of the two panels here, the one that's labeled PTYR is a way in which we can measure how active MuSK is as an enzyme. And the blacker that line is, that signal is, the better or stronger the enzyme is. And so what I've shown you on the far left, you can see agrin. And so these muscle cells, and remember I told you if you treat muscle cells with agrin, it'll bind LRP4, which will bind MuSK and stimulate MuSK. And so you can see if the cells have been treated with agrin for 10 minutes, there's a little bit, a gray little black line there. So there's a little bit of MuSK. It's agrin is stimulated MuSK. And if I leave agrin there for 45 minutes, you can see that line's become stronger. So the longer agrin is there, the better chance it has to bind to MuSK and to LRP4 and stimulate MuSK. And that's that increase in that black line. And the numbers that you see above, 13, 21, 22, G-GD, these are different antibodies that recognize MuSK. And let's focus on 21. 21 is an antibody that binds MuSK very well, but as you can see, no black line at 10 minutes nor at 45 minutes. It does nothing. It doesn't antagonize MuSK, doesn't inhibit it, and it doesn't stimulate it. Whereas the antibodies labeled 13 and 22 are very strong stimulators of MuSK. At 10 minutes, there's a very strong signal, as strong as you see at 45 minutes. And the same for the antibody that's labeled number 22. So these are examples of antibodies that bind MuSK and stimulate the kinase. So these are exactly what you want. They're molecules that are agonists. They can stimulate the kinase. So these are simple studies that are with muscle cells that we can grow outside of the animal. And we've just begun a long series of studies together with Genentech to use these antibodies in a mouse. And the first thing you'd want to do is to introduce these antibodies into the mouse and then ask, is MuSK more active in vivo in the mouse when these antibodies are present? And if so, you need to know when they become active, how long they're active, what's the best route, what's the best dosage, et cetera. And so these are a long series of studies that have just begun over the past few months. And I must say it's the first time I've had this kind of extensive collaboration with a biotech company, and it just couldn't be better. It's a wonderful, wonderful collaboration, a lot of interactions. We talk regularly. Fortunately, we haven't had these kind of problems with our Skype conversations, so that they've run relatively smoothly, and we continue to exchange information, and we're starting to move forward. It's going to take us some time to get this experiment complete in the mouse, but we think this will tell us a lot. So we'd like to know, can these antibodies stimulate the kinase in vivo? And then can they do what this genetic trickery did in the mouse model of ALS? Can they keep the nerves attached to the muscle longer? Can they improve motor function? Can we manipulate the system with the antibodies the way we've been able to do by genetic trickery? And I'm certainly, this is a very early stage, but you can be nothing but hopeful, because I think we had a very large surprise in the past year or two with these studies that I did describe to you to show that by increasing this retrograde signaling from the muscle back to the nerve, we actually can keep the nerves attached to the muscle for a long period of time and improve motor function. So I'm very hopeful of these studies. So I'm sorry for the delays and the time it's taken me to go through this, but I appreciate your attention throughout the talk. And I'm be glad to answer questions.
We are here. I'm waiting for Lucy, who might have gotten accidentally muted when we had the audio issue. Just a moment.
Hear me?
We hear you now.
Oh, excellent. I will repeat that. Thank you very much, Steve, for taking the time to really explain that very, very carefully. It's absolutely fascinating work and well illustrated with just seeing the imagery of how these synapses attach. And also, I think, so encouraging that you really can make partnerships with biotech and academia to move these fascinating findings to something that could be more therapeutic. Because of time, I'm only going to take very few questions. We actually have up at the moment only 2. I do encourage you, if you have additional questions, to send them to the association. We will certainly get them addressed. There may have been some that joined late that might have experienced some audio problems as well themselves. I'm very sorry for that. We will look into what the issues were and why Steve had challenges as well. But for those that hung in and could follow it, I'm very grateful. I'm going to take the 2 questions because both are extremely relevant, very different ones. One came very early to say, is there any work being done to diagnose the disease early before onset by using this information, see that you presented that there's this breakage from the synapse, obviously diagnostically in humans, and can that be detected in the near future in humans? So maybe you can comment on that, Steve. It's a little outside of what you're doing at the moment, but your thoughts?
Yeah, I guess I have 2 thoughts about that. One is that certainly for sporadic, you could biopsy muscle and ask what percentage of the synapses are denervated. And so that would be an early indicator of ALS if you saw extensive denervation. It could even be 10, 20%. It wouldn't produce clinical motor dysfunction signs, but would be indicative of disease. But again, obviously you wouldn't do that on a routine basis, so that would not be applicable for sporadic ALS where there is no sign or indicator of disease without initial symptoms. In cases in which there's a genetic cause of the disease, in which I think there are better ways to diagnose the disease at an earlier stage where you can expect or anticipate that there may be disease symptoms. There are genetic ways in which you could identify and whether or not a family member carries an SOD mutation or a chromosome 9 mutation. So I don't see this as a very effective diagnostic tool. Now the question is, are there other ways in which one could signs of denervation at still earlier stages? And I suspect Lucy's in a better position to answer that than I am. I know there are a lot of studies ongoing to look for biomarkers that might be in the serum that would be, much simpler to assess and measure than doing measuring biomarkers by muscle biopsies, which would be quite intrusive. So
I think you captured that correctly, Steve, and I think that maybe there will be markers that align with the concept that there's denervation, changes in phosphorylation of certain things, but certainly biopsy is not a sort of a routine thing to be done. And I think what it interestingly leads on to the next question. The listener asked, well, it's all well and good, these are wonderful mouse experiments, but how can we know if it works for humans? And I think that's the trick of translating and measuring and using humans as a step. Obviously, this has to be done in the mouse first, but interested to see how you would comment and what the next steps are to make sure we can move this into treatment and have relevance for humans.
Right. So I'm very hopeful that I made the point early on. I guess there are several reasons why I'm hopeful. One is that this synapse, whether it's in a mouse, whether it's in a rat, whether it's in a frog, whether it's in a lizard, is very, very much the same. It works the same. It looks the same. The molecules are all there. MuSK was a molecule which we first discovered, it's nearly 20 years ago, and we first discovered it in a fish. And then we went on to show that it was in mouse and then in human. Acetylcholine receptors, that's in fish, chicken, birds, mammals. LRP4, this molecule I described, not only is it present in all of these synapses, it's a very large molecule. It's made up of hundreds of amino acids, and there are very few that are different. It's 96% identical from human to a fish. So there's tremendous conservation in all of the molecules. There's tremendous conservation in the way the synapse looks and works. And when you look at the individual molecules, they're very much conserved throughout evolution. So I think in that perspective alone, there's a lot to be encouraging about. And the other is that my view is that in instances where therapies that work in a mouse don't work in human, I suspect that many of the cases are not due to the fact that the underlying biology is different, but rather that the molecules that are used or the drugs that are used just are metabolized and handled differently in a mouse than in a human. Now, and that would be more common with small molecules than would be with antibodies. So I think this particular approach of the antibody approach, I think is less likely to run into the types of problems that one sees with different metabolic behavior in rodents than in humans. So I'm very hopeful for that. The other reason I'm somewhat hopeful is that, remember, there are many forms of ALS. There are sporadic forms and there are many genetic forms. So in the genetic forms, if you decided that you wanted to cure things that are due to altered forms of SOD1, that would be pretty wonderful. But it would leave a lot of people with other forms of ALS without any effective therapy. And that would be true for any one of the genetic diseases. And then you'd have the sporadic forms, again, that would be without any effective treatment. This particular perspective of keeping the nerve attached to the muscle, this is a part I made early on, is that all forms of ALS, whether they're the genetic forms and all of the genetic forms or the sporadic forms, they share this loss of motor innervation. So we think we're tackling a part of the problem that occurs in all forms of ALS. So if it is successful in the mouse, and if it proves successful in humans, we think it could have widespread utility in all forms of ALS, and that is appealing as well.
I think that's exciting, and I would just quickly add before we end that you illustrated so well that the synapse is an early problem and needs to be maintained, but certainly it would be even better if we could at the same time maintain the motor neuron. And if we think of additional approaches we're taking to maybe through stem cells enhance the milieu of the motor neuron through GDNF and other factors, I think this lends itself incredibly well to a combination approach as we try and approach helping the motor neuron itself survive. So I think that you illustrate very well that this is something that can be translated. I'm encouraged by a partnership with a company who has been successful in moving in many disease areas treatments to the clinic, and I think that this is a very promising approach. So I'm sorry to those who might have wanted to ask questions, although I'm not seeing others coming in. I thank you again for your patience. We usually are very rigorous about keeping to time, but I couldn't resist to make sure we saw and heard the complete presentation. And again, thank you so much, Steve, and thank you for your patience in fiddling around with the technology.
Thank you very much. It was a pleasure.
Thanks all, and next time you'll be listening to me, I hope you can hear me clearly next time, and we'll hopefully solve the audio challenges. And the next will be in July, where I'll give a broader overview. So looking forward to hearing from you again then. Thank you, Steve. Thank you all. Good night.
Thank you very much.